installation and move. Matthias? Oh. No, I think I just need that you unlock yours. Did they give you the second presentation? No, I'm confused.
I copied just one of yours.
Okay. You can get the one from Git maybe? Oh, no,
Oh, should I can take it from the Git. Which one is it, Ivan?
The introduction footprint. Das ist auf dem Desktop kopiert, oder? Meinst du?
Nee, nicht. Ich habe nur das eine kopiert. Also, warte mal, das ist die... no, sorry, das ist...
Genau.
Slides.
Mach mal die Namen weiter.
Ah, Mann, ey, jetzt ist er schon wieder zurückgesprungen in das andere Verzeichnis.
Nein, ISMB to tun.
Genau. Hier,
Genau,
oder?
Okay.
So, Ivan, PDF, okay?
I think it's passing automatically. It was passing automatically. Can you put the because you put slideshow, right? You have to put the
Full screen just.
full screen. You have everything in German, huh? Yeah, okay. Should be better. Okay, so we can start now the second part of this tutorial. We have a little statistical question now. So how many of you were able to finish the first part? I mean, to do all the exercises? Okay, so most of the have that's still here. And how many were able to install all the tools so far? Okay, not many. Because of course, we can have now 2 tools to install, but I think we will be able to keep things in track. Okay, so this part now I'm gonna introduce the analysis of footprints, so to analyze open chromatin data to find transcription factor binding sites. And then next Marcel will talk about regulatory networks, and we're gonna do the practical part together. So the practical footprints and regulatory networks are gonna be as one. You have seen in my first presentation that we are interested in finding chromatin, and the important thing about chromatin is that you can learn, for example, where, given that we know the particular histone modifications are usually indicative of active either enhancers or promoter regions, then they can help us to identify regions which are potentially bounded by transcription factor binding. And this, if you can now measure ChIP-seq data, as we have shown in the first part of the practical, you can really now identify either enhancers or active promoters in cells. And the other part, there is other protocol which I show you, which is called Dnase, which is originally done to measure open chromatin, but it has this nice feature that, given that the cuts of the cuts of the Dnase is actually recapitulating regions which are open, if you do the right counting, you will be able to get data, this kind of footprint-like structures, and these are actually the regions where transcription factor bindings are sitting. Meaning that with one experiment, you could, in theory, find all potential binding sites in one cell. What we have been working on, and this is the tool which you're going to show today, tries to explore this, that there is some sort of grammar, and this grammar is kind of depicted here, which means that first you have the grammar of the footprints inside the DNAs, but a similar thing also happens in the histone. So in regions where you have a lot of transcription factor binding, you should not have histones, but the histones, in particular the ones which are associated to active regions, they should be accumulated before and after, maybe in different levels, for example, more here than here. But you also have a kind of a depletion or another footprint. And so you can now think about segmentation models that look genome-wide and try to find this kind of values between DNAs and values between footprints. And this is actually what we have worked in the past few years. So we have proposed a method which is called HINT. It's a hidden Markov model-based model that you can either give the DNA's histones or both of these signals, and out of this, we'll try to predict either these large histone footprints or the small DNA footprints. There is a lot of deal of computation involved in this. I'm going to show a few of them. So, of course, one is the model itself, but actually before you start with the model, you need to do a lot of normalization steps. I'm going to show one of the details, which is important for DNAs. And one important thing is that when we did these models, we were able to build a more than one cell type, but apply to any other, showing that somehow this chromatin grammar is independent of the cell. So it's something which is universal. Okay, so how does the model work? So, okay, so this is, I think the animation went wrong here, but the idea is that you have HMM with 8 steps, and you have one state here which is indicated in the background. In the background, you should have no signal of histones and DNAs. And the idea is that before you reach the footprint, which is what we want to identify, you need to first see an increase in the histone and a decrease in the histone levels. And to be able to do this, what we include is a path, which is, so before we want to reach the state here, the footprint model, but to do this, we have to first see an increase in the histone levels. And then once we do this, we want to see now an increase in the DNase levels, for example, indicated now by these states, so the DNase level states. And only then, we will be able to go into the footprint. And once you're in the footprint, you can see many peaks going up and down. So you're going to have many footprints one close together, indicating that many transcription factors can be binding close. And when the histone levels start to go up, you would go outside of the DNA states, and you could, of course, either go back here or go back to the background, which is modeling this increase now in the histones. So this is just a way how we can use a generative model histones to model this grammar. And in this case, we really did these models by hand. We annotated one region by hand, and we trained it with maximum likelihood, and this was actually working quite well. Of course, there are some tricks. For example, to be able to model the up and downs, you don't only use the signals I'm showing here, but we also calculate the slopes of the signals, because the slope then will help us to identify the ups and downs. And usually this improves the model overall. But there are many artifacts, in particular for DNAs, and this is something that actually was a big discussion in the literature of DNA analysis. And this is just a very simple scheme of this problem, and the problem is like this. So DNAs, or an enzyme that is used for this protocol, they have a preference to bind to particular DNAs. And here, for example, we know that DNAs binds better things which have a CG in the middle than things which have a AA or AT. And for example, if we look at many libraries of DNA-Seq, what we're going to see is that we usually find 3.5 more time reads which are aligned to these schemas, where the read starts in the middle of the schema. Then we find these schemas in the genome, so frequent-wise. And indicating that for some reason, DNA likes to cut more these sequences. Of course, if you do the same statistics for these red guys here, you should see a value below 1. So, and this is actually reflected by this thing here. So here, what we have is we have ChIP-seq data for one particular factor, EGR1, and you see we see the clear motif of EGR1. But if we look at the uncorrected DNA signal, you see not a footprint, which is only a value, but you see an increase. And this has to do with the fact that these regions are conserved, are CG-rich, and DNase likes to bind to CG-rich, and then it creates an anti-footprint. But if we do a simple correction, which is just multiplying the counts that you see by this bias correction, or dividing, sorry, so you're kind of imposing down if we have a higher bias, and you would be able to create this footprint kind of level, which actually is behaving exactly. And this is just an example how the experimental protocol can really have a high impact on how well you can predict the footprints. So how does it look in reality? So this is a real example of K562 cells. So this is a promoter of a gene which is being expressed. And you see here there is a lot of histone atrial methylation in the promoter, which is indicative that it's being expressed. And also you see that there are kind of two values, and in these values you have the high DNase content. You also have a kind of enhancer region, which is supported by this monomethylation, which also has a lot of high DNase cuts. And in the end, the footprinting method is only predicting regions like this. So everything you do is just filtering your genome and saying in these cell types, just look at these regions for binding sites. And that's it. But of course, you want to know more, who can be binding there, and then you can go back now to classic motif match analysis. Here's, for example, the motif match for 4 particular motifs. And now you can ask things like, what are the motif matches which have a good overlap with the footprints? For example, CTCF binds has a high overlap here and here for these two, but not for these two. And this, the meaning here would be, maybe these are real CTCF binding sites, but not in this cell. But these two are, at least from the chromatin data, supportive that they are functional here. So the point here is, how can we evaluate this? And the nice thing about this problem, in contrast to the ChIP-seq data problem we had before, is that it's easy to evaluate because we have ChIP-seq data for some of these factors. For example, we have ChIP-seq data for CTCF, and now we can see that whenever we have a footprint which was supported by a motif match and also by a ChIP-seq peak, this should be a true positive, right? On the other hand, if you don't have a peak, but also you didn't have a footprint, so it means that this CTCF shouldn't be active here, and this should be a false negative. And then, of course, you can have also the false negatives and false positives, and out of this you can do AUPRs, IUCs, and evaluate methods. Of course, now not looking ChIP-seq for one factors, but hundreds of factors that you can get in code. And this is an example of evaluation of different methods. I'm not going to go into details of this evaluation, but just to give ideas of how the different information can improve how you evaluate. So here is the UPR, so 100 would be best. And this is for 88 factors in 2 cell lines from ENCODE. This is what happens if you only look motif match. So it does quite bad for most of the factors with very few exceptions. If you do the footprints on the histones, you improve much more, but it's still not so good. But in some cases, if you only have histones, it's better than only looking at the sequence itself. This is only, tag count is only counting how many reads you have around your footprints. This is the easiest thing you can do with DNase. Again, better. And these are many tools that people has been proposing in the past years to try to do footprints. And this is what we have been working on, only using DNase and combining DNase and histones, so we can further improve a little bit. And this is just showing that actually we can really improve a lot the prediction power by combining different data sets. So if we have chromatin data, this is really useful for finding binding sites. If you are curious about which histones to use, this plot is a little bit too busy. I don't like this, but what I'm showing is only I get all combinations of activation marks, so 1, 2, or 3 combination marks, combining with the DNase. And we see that, of course, overall, using more histons is better, but it's not really that, let's say using 2 histons are usually almost better than using 3. But the top scoring actually typical histons that I have shown you in the first slide. So A3K4 monomethylation, A3K4 trimethylation, and A3K9 acetylation. So using the ones that people have been showing in the literature are the ones that actually can help you, but many combinations do equally like. Okay, so I'm almost finished with this part of the footprint, but one issue is that we have been talking a lot about the Dnase, but in the protocol, in the tutorial, I'm going to show how to evaluate ataxic. And ataxic is a protocol that actually does almost the same thing as the Dnase. The difference is that instead of using the Dnase to cut, it uses this TNF5, which is, works in a little bit complex way. So it's a dimer, inserts adapters here, but in the end, it's fragmenting DNA where it's open, and you can get the fragments and sequence them. And if you look at the beginning, this should be the regions where you have open chromatin, just like with DNAs. And there is a little bit advantages why ataxic is now starting to be adapted in the literature. So is that it works with few cells. So it's easier for the experiments to do. It also takes fewer hours, so you can do it in half of a day, while ataxic takes up to 1 week. But the price is that it's TN5s, they are larger proteins, and so overall the cleavage cuts are not so good. And maybe the explanation is that, for example, if you don't, by being a bigger protein than the DNAs, you will not be able to cut many sites around binding sites which are close together. But this is something that actually is still kind of open, and people don't have a good solution. But we have been working on this in the past year. So, for example, this is just a comparison now of these AUPRs of same factors using DNase and attack. What you can see is that, of course, DNase usually is doing better, but it's not much, I mean, the ataxic is not doing a worse job. And for some particular factors, for example, this is the profiles, every various profiles, in particular for P1, for some reason ataxic works better. We don't understand this. So TCF, for example, the motive for ataxic is not so good. But at least what I can show is that ataxic do find footprints. So this is just an overview of the result of this part. So I'll show you how we can use footprint to detect transcription factor binding sites specific to one cell. And the cleavage correction was crucial. And that the same thing can also apply for ataxic, which is something that's experimentally favorable, and is a protocol that's starting to be more and more adapted. And we have just released now this to work this in this part. Okay, so I think this was for this part. So is there any particular questions for this part of the talk? Yeah. So? Yeah.
I have a question about the slide where you were showing different methods as a box plot.
Okay. Yeah.
So on that slide, the only method which uses the sequence was PWM. Everyone else was using only the RNA.
The Dnase.
The Dnase, sorry.
Yeah. So I will have to look at details now. This paper is already more than 1 year old, but I think Centipede, for example, tries to use the sequence. So some methods try to use sequence information, sometimes more larger features like CG content. I don't remember now, but if you look in the paper, we have all this kind of information there.
But my consideration is that if you use motifs as true positives kind of, can't you have the situations where methods which use sequence information, they have higher scores because they also use sequence information, or because they're better?
Yeah, sure, you're right. Yeah, but I think most of the methods are not using motif match. But there isn't actually in practice. I didn't really show the difference about the methods. But the method that hint works, it's kind of segmentation. So we really don't look at we really scan genome wide, and this is the case for Wellington and Boyle and Dean and Peak. Yeah, so half of these methods are doing this, but other methods are more machine learning driven, so that they start with motif matches. They look at the profiles around these motif matches, and then they do this usually per factor, and they try to train. And what we have seen in the evaluation that, of course, for some factors, some of these methods which are factor specific are better. But at average, the models which are segmentation-based, which are really not even looking at the factors around, or only looking regions with a particular factor, do at average better. And I like them better because from the perspective, usually we don't know what we want to do, and I also want a model which is less biased towards one particular factor. But of course, the cleavage patterns differ by factor, also because of combining patterns, and it's still worth to look at this at some point. And we also show how to do this in the practical part. Yeah?
Yeah, so you said that TF5 use-grounded sequencing is rather big, but then isn't this insertion, like the cleavage, isn't that going to be quite a bit from like the boundary of the prescription factor with the native DNA? And do you correct for that, or do you?
Yeah, I think, so the status of TAC-seq currently is this, that this has been published, 2013, so it's almost 4 years old. And people are, for some reason, not really doing footprinting there and not really looking at these details. This is what we did, and yes, it's important that because the protein is bigger, means that the binding cleavage size is not going to be it's going to be maybe far away from the next protein. So you need to correct for this. And this is the things that we are currently working on, that we will try to actually we are currently writing up this part. So looking at these things is very important, and something that for some reason people were not carefully looking at the literature yet. And of course, Dnase will still be better than a TAC-seq, at least that's what we see. In our evaluations, everybody has seen it before, but experimentally it's much more favorable. You will not be able to do Dnase in this hematopoietic system that we are going to analyze, simply because you don't have enough mice to have the number of cells. This will be too expensive, or yeah. No more questions? Okay, then I think Marcel can follow up.
All right, so now that's the last part of the lecture, so to speak, for the tutorial. We're going to talk about regulatory networks. Actually, I was going to talk about the differences for the TF annotation. That was one of the questions. So maybe, hopefully it's becoming more clear. I'm Marcel Schulz. I actually work in Saarbrücken in Germany, also at the MPI for Informatics. And Florian is also my student, who's also helping you later in the practical. And so remember, as we said in the beginning, what we're interested here, and what you saw in Practical 1, we're trying to understand how, from precursor cells, these differentiated cells are made in our bodies, and which factors are driving these changes, right? In particular, which transcription factors. And so you already heard, and maybe some of you are also familiar with this, that on the left side, there's different experimental methods. The ChIP-seq you can do with TFs or protein binding microarrays and with SELEX. And these are all very costly methods, and they're often limited, and you would never really do them for all the TFs, even for one cell type. And so what we're talking about today is on the right side is these computational methods, and there's two different types of methods. I'm going to introduce them. One will be site-centric, and another one will be segmentation-based. And we're going to use position weight matrices, as most of you may be aware of, where we just have a bunch of sites of a binding sites of a certain factor, and then we're going to look at different positions. We're going to just ask which nucleotide is preferentially found there, and then you can visualize this with sequence logos, like here for CTCF, where you see that at some positions, the factor really prefers to have a C or a G, and in others it's much more flexible. And it really depends on the factor that you have, how good the description of this is, and also how good you're able to find genome-wide matches for this, even with epigenomic data. And so, sadly, depending on the system you're working on, it will depend more or less on the quality also of this type of binding data you have. These PWMs also, they assume independency of positions, which is a simplification. And there are other methods that are, able to model dependencies between positions, but most of the databases where you can really download a lot of different TFs for doing this kind of annotation that we're talking about are still based on PWMs, and I hope that actually as a community we can improve this. So what are these databases? So Transfac is a commercial database, and Jasper and UniProbe and Hokumoku, these are large databases for example, in Jasper for different species, and in hundreds of factors you can download these PWMs and you can look at the preferences of binding for any of these proteins in the DNA. And so we compiled the data set that's part of the T-EPIC software that we've developed that basically, non-redundant set of hundreds, over 500 TFs for mouse now, what we're going to use. And so, classically, what are the approaches to how to use these PWMs, or these motifs, as Ivan also called them, right? So you would take the genome, you take a sliding window, you have this PWM, and then you, given a statistical method to compute scores with these probabilities, you just annotate it. And so these are the highlighted regions here, which are sort of like the reported binding sites, right? It's just sites where the sequence is very similar to this consensus site, as you saw in the motif, like for CDCF, the positions where you have the C's and G's clearly there. And so, you can easily do this. But then, of course, if you look in an individual cell type, you will have the situation that some of these sites are actually really true binding sites and others are not, right? They're just wrong. And why are they wrong? Well, if you look at the whole genome, it's really too large of a sequence to look at. Just by chance, we'll have many false positives because the motifs are short, right? But even if that was not so much of a problem, the truth is, of course, not in all cell types all of these binding sites are accessible even to proteins, or for some reason these proteins just don't go there. Maybe we don't even know what the causality is, but they're not sitting there. And so this is because the genes are, they're in heterochromatic regions, so they're not really accessible. Like not even a gene, any region in the genome really, like an enhancer or so, is not accessible. And so if you would just scan along the sequence, right, you would just simply not be able to say if it was sitting there or not. So you need this additional information. This is the epigenetic data that we're using here. And so, so in principle, how this method works, like also some that even just you mentioned, like PAQ, a centipede, right? So that you take a motif and then you annotate the genome, and you get these regions, right? And now, if you have the epigenomic signal along this, for example, like the Dnase, let's say, right? And you can look at peaks, and you know how to call peaks now, and in these regions of the genome where you have the motif and you have a peak, or you have high signal determined by some statistical model, you would consider that, okay, no, this is an actual binding site, right? And the other sites where there is no such signal, well, it seems like the DNA is not accessible, and so it's very unlikely that actually this particular CDCF or whatever factor you're looking at is really sitting there, right? And so that is the basic information that we're trying to use here, that we have a cell type specific assay. It's going to telling us where a DNA binding protein can be sitting, and these tools, among others, are trying to integrate this information to generate a cell type specific prediction where transcription factors are binding. And so now the site-centric method, that's why I call them site-centric, they start with the site and then they overlay the epigenetic data. Okay, so the alternative way is that you use a segmentation-based approach, like what Ivan also explained, but it could be others, essentially where you're just trying to call peaks with the data, so completely data-driven, and depending on the assay you use, you find regions where you have a peak, right? And then you use methods now to basically in these peak regions, you try to ask, okay, now which factors are sitting there? And so for the tutorial, you're actually going to use another method. It's called TRAP. It's based on a biophysical model where you also use PWMs, but you're not trying to find a particular site, but you're just asking, well, in this region, let's say 150 base pairs, how likely is it that a factor is sitting there? And so this TRAP model uses a biophysical approach that tries to model the energy landscape and tries to say, like, how many sequences are very likely to be bound by that factor, and so you get you can think of it as the expected number of TF molecules that are sitting there. And so basically, what you get then for each of these peak regions for all the TFs, right? You get these rational numbers, and the higher they are, the more likely is it that the factor would be binding this region. Good. And so why is this necessary? You may ask yourself. Well, obviously this motif annotation or the site-centric view is based on some statistical model which does not necessarily have anything to do with what's really going on in the cell, right? In particular, there has been works been shown that this idea that a certain site is or is not bound by a factor is also not really realistic, right? Because probably the protein is going along the DNA and then with some affinity staying longer or not so long at certain genomic regions, and then depending on other factors, it may be staying even locked in there or staying there for longer. So this binary idea is, I guess, a simplification that we just like to do, but it's not super realistic. And so that's why we're using this trap method. But just that you understand what's the difference, right? So if I use a site-centric method, I will basically find positions in the genome where I have a motif match and I have a peak of my epigenetic signal. And whereas in the segmentation-based methods where you use this biophysical model, like I explained, you're going to have, for all of these segmented regions, you're going to have a list of factors that may be binding there, right? Because it may not just be one. So now that you have this data, how can you now link this to changes of gene expression, right? That's the question. And so what we'll be using now in tutorial is a software that we developed, and we applied this to many systems already, which we call Dynamite. And it basically answers the following question, right? So you do a gene expression experiment between case and control, and you find genes that are upregulated, downregulated. So now you're trying to understand, which are the regulators that have been driving these changes. And so, here's 3 different transcription factors shown. And for example, you can see that it seems like the red TF is preferentially binding upregulating genes, so it seems to be activating those. Whereas in downregulated genes, there's the other TF3, the pyramid binding, which seems to be like repressing these in the cell type. Or has been active in the other thing that you compare to. And so now remember what, the task here, right? So we're looking at hematopoiesis differentiation. And so we have the precursor cells and the differentiated cells, right? And so, for example, upregulated genes would be like, let's say, cells that have higher expression in differentiated B cells, but they are otherwise downregulated in the precursor cell. So something that the cell somehow selected for to making the phenotypic difference of the cell type. And so, so now in order to do this, right, so far we basically just called regions everywhere in the genome, right? But now we're looking at genes. And so we need a way of summarizing the information, projecting them to genes. And the way that we're doing this is like we take the transcription start site of each gene, which is known here as a TSS. So we have I is the gene, and we have the transcription start site. And then I hope you can see these red parts, which are labeled like G, J1, G, J2, and so on. These are the footprints, for example, or peaks that you found in the vicinity of this gene, all right? And why are we looking also, we're looking in a larger region. It's like a window of 30 kb, because as we said in introduction, it's sadly so that not only proteins binding directly at the transcription start site, but also factors binding much further away, like in so-called enhancer regions, are probably influencing the gene. And we and others have really shown that this is quite important to take that into consideration. And you can easily verify this also in the tutorial. You could just change these parameters and you would see the differences. And so you really sort of like need to at least make some window which is larger to incorporate these regions. And but because this window is quite large, what we do in practice is that we put an exponential prior on this. And so the further away that region is, the less likely it is to influence the gene, right? It's still possible, but it's less likely. And so this is shown in the formula now. You take the contribution of a certain factor in each of these regions, weighted by the distance with this exponential decay. So at the end, what you're going to get is a score for each gene and each TF, where you sum the basically expected number of molecules sitting in each of these regions. Yes?
The form of this fall-off is just completely arbitrary.
It's completely arbitrary, yes.
You have no justification for it.
Well, the justification is the further away it is, the less likely is it to influence the gene. But you're totally right. If we had other data like high-C data, we would be able to do better, but normally you don't. And actually, even if you have it, it's not trivial to do this. That doesn't work well. If you put a uniform prior, so to speak, then it really doesn't work well because there may be other genes in the vicinity, and then if you take their regulatory regions with the same amount projecting on a TS much further away, then basically you're introducing noise.
Right, but it might just be right also. I mean,
Yeah, and we tried it, and others tried it, and we can easily show you that it doesn't work well.
We know there are enhancers and megabase upstream in terms
Yes.
of G.
Yeah, and so.
It seems like we've got an arbitrary window and then an arbitrary
Yes.
format, which is going to then result in some arbitrary result.
No, it's not arbitrary because we know exactly what we're doing. But the point is, if you want to include a megabase region, it's really there's too many open sites that you will have to take into consideration, and you just don't know which of these really linking to the gene. So sadly, if you want to do this, you need just much more data. Yes. I didn't. Other people did, but I'm not saying that this is the perfect distribution to use. Okay, so anyway, so what we're going to get for each gene is a score that you can think of this as the likelihood or the number of factors that are probably binding and influencing this gene. Good, so now that we have this, we get these, what we call affinity matrices, yeah, so because this quantity is called the affinity in this trap tool, and so for each gene and for each TF, we're going to have the affinities, right? In some ways it will be high, in others will not be high, and we're going to have this for the two cell types, so cell type 1 and cell type 2. And so, as I said, we're trying to find now the genes where a certain factor is, you know, only sitting in cell type 1, but not sitting in cell type 2, for example, right? Or sitting much more, with more copies maybe in cell type 1 and cell type 2. And so what we're doing is we're doing the ratio of the two values, right? So we take TF1 in gene A in cell type 1 divided by TF1 in gene A in cell type 2. And this gives us this ratio matrix, which denotes the difference of binding likelihood of a factor for a gene. Good. And so this is like the overall workflow that you can use, right? So the OP chromatin data is, you know, you call these segments like the footprints, what Ivan explained. Then T-Epic is a software that does this annotation with trap, where you get for all the TFs, you know, what's their affinity, and it also summarizes on the gene level, so you get these matrices per gene and per TF. And then you can create these ratio matrices I explained, and then if you have a set of differentially expressed genes, which we provide to you, you can link them with the software Dynamite. Basically what Dynamite is doing, it creates, it builds a classifier where you can think of this, right, we have this, this matrix I just explained for gene to TF, and now we want to see which TFs are linking to certain classes of genes, either up or down regulated. So we treat it as a binary problem. You can also do regression, but here we're going to do classification. Yeah, so how are we doing this now exactly in the tutorial, right? You already did the p-calls, so differential p-calls are available, and you will also do footprints. And so one particularly nice way, if you have all of this type of epigenetic data like we do have now in this tutorial, is that you can call the footprints, which you see on top, which are these regions where it's very likely that a factor is binding, right? Then you can do differential p-calls where these are regions where, you know, one cell type compared to the other, really something is changing. And if you overlay the two, then you're really concentrating on these accessible regions, at least in one of the cell types, that are seeming to be important for making the difference of both of them, right? And so, yeah, so this is one of the steps in the tutorial, and so this is, you can think of this as the input. But I'm going to say that, you know, this kind of strategy also works if you're not doing the differential p-calls. You can also just make the footprints in both cell types, you know, run the annotation, and then run the tools. And you can run different assays. You can use DNAs, or you can use ATAC, or you can just use K27 acetylation. We tried it with all of them. You know, you will always get, you know, a piece of information, but obviously there are differences in how the assays work, and so you're going to see that. All right, so how is it, you know, I'm showing now one example here to give you an idea. So let's say we look at this precursor LSK cells versus B cells, and then we have now with the footprints, these are the numbers that we have available, that also in your local folder. And we, you know, got differentially expressed genes, which are 2,200. And of these 2,200, actually 466 have one of these differential peaks in this 30 kb window that I talked about, that we use for the annotation. Okay. So basically, right now, what we're doing is, and also because it's faster, and so you can run it on your laptop, we're going to look at just these 466 genes, and then going to try to find factors that explain difference in expression for these. And so this is what the Dynamite software does. It uses a logistic regression classifier that you can interpret, where you get weights for each of the TFs. And so how good does it work? The software will provide you a plot like this, where on the y-axis you see the value between 0 and 1. It's for the performance of the classification, and it gives you different metrics. I think the easiest often to look at is test accuracy, which is the third. And you also see an error bar, which is like with basically cross-validation, how it differs. But in this example, what you can see that for these 466 genes, our accuracy is really high. So with good accuracy, we're able to finding transcription factors that explain difference in gene expression in both of these cell types. Yeah? So when you're using this kind of approach, right, so what I really like about this, the accuracy is telling you how good you are doing. Yeah? So if you're doing, let's say, less accurate with, like, let's say 70% or so, it doesn't necessarily mean that your data is crap. It also doesn't necessarily mean that you don't have the factors that you need. But there's a lot of noise or biases that go into this this kind of analysis. Obviously, we're combining different types of data. But one really thing in practice that I see also is often leading to not so good accuracy is when you have patient data, yeah? And the people are much more heterogeneous, so if you measure epigenetic data for them, there's much more variance, and so the way that we're putting this together here, then often it's much harder to really explain differences just from the TF binding in these open chromatin regions. But this is mouse data, and this is also really good data, that's why we chose it, and you see that it works really well for separating the expression of these gene sets. All right, and so another thing that you get as an output is this heat map, which is telling you which are the features that the classifiers picked for making these decisions. And actually, each row is a different fold of the cross-validation, so you also see a little bit, if I take different subsets of the data, does it change a lot, right? Which transcription factor seems to be associated with the changes. But most of the time, it's very consistent. And so now each row is a transcription factor, except for the first two rows, which are peak length and the peak counts. And you see that some factors are seemingly very important for explaining the difference between these two. And so that's very good. This already gives you an idea which factors are really relevant for explaining differences in gene expression. And because now we have a lot of data, right? We know the segments, we know which TFs, we have their affinities, we know if they're binding there, we can actually dig much more deeper if we find some of these factors interesting. For example, the HLTF factor here is one of the most strongest features selected by the classifier. And so you can run another module in the software that gives you a detailed plot just for one TF. And so this is what is shown here. On the right side, the heat map is basically just what I explained before. It's just like a size of this. And so now what you see in the middle is, I think it's better to concentrate on the middle now. We see the complete densities of these two cell types. So there's the darker one is the precursor cell type, and the B cell one is the more light one. And we basically show for all the 466 genes these transcription factor affinities that we measured in these two cell types. And you see that the densities really look different, right? So this factor in B cells is binding, it seems to be binding many of these genes that are differentially expressed because the affinity, so the likelihood of binding is much higher. Yeah? Whereas in the precursor cell is very peaked on low affinity binding sites. And you can actually, below, you can see a scatter plot where we directly compare the amount, it's a predicted number of molecules for this HLTF factor in B versus T versus the precursor cells. And actually, the color coding now is really important because what the shading is if it's over or underexpressed. So the red would be overexpressed and blue would be underexpressed. And what you see here is that this factor is actually whenever it's highly, so there's a lot of binding in B cells, yeah, then this gene is higher, like shows more expression in B cells. And because we do a ratio of B over LSK, then see this as a blue color. And the other way around, if it's more binding in LSK cells, then there's higher expression of the gene in LSK, right, in the precursor cell type. So which means, together with this information, you can conclude that this is an activator. Whenever it binds, it leads to more gene expression, and there's regulation going on that the regions that are bound by this factor in the cell differs between the two cell types. And so this is really helpful if you want to plan, experiments to validate anything of this in detail. All right, so now you've seen the method. In the practical now you will learn how to do these footprints and then use dynamite, and if you have questions, then I'm welcome to answer them now. No, no. It's well, we also have the peak sizes. Different. This is one of the features, just because that's a confounding factor. Obviously, if we have much more peaks, then that also gives us some information, right? But we also want to obviously understand which TFs are driving these changes. And so this is the step that I explained here. So we're going to have, so we have all genes. We look in the footprints that we find in the vicinity of the TSS, and each of these footprints we annotate with the likelihood that the factor is sitting there. We sum up these values weighted by distance. And so for each gene, and now I do this for cell type 1, cell type 2, and if this number differs a lot, then it seems to be an interesting feature, right? It seems to be that suddenly the sites where the factor could be sitting before are not accessible anymore, and probably the factor is not sitting there. And so this is exactly what is encoded in this matrix. Yeah? So each of these numbers. And so we have these two different cell types, we do the ratio, and this is what goes in as a feature for the classifier. No, cell type 1 and cell type 2. So these are the two, it's, you know, the arrow gives you the ratio between both cell types.
Ah, okay. So it's the ratio which you use as an input for the classifier.
Exactly.
Okay.
Yes. Can you speak up, please? Yeah. Mm-hmm.
So that means that you need to have these chipset data for all TFs to
No. ChIP-seq is not for the TF. As I said, we're not using data for.
But I mean, you need to have these data sets. This is what you have used in order to identify the differential regions that you have done the first step.
Yeah, you need at least one epigenomic assay, Dnase, attack, seq, histone, anything like this you need to be able to run Dynamite, yes. Yeah? But you don't need a combination of them necessarily. I mean, as I said before, it's often helpful, and also even showed this, right? It's really helpful if you have more of them because they are somewhat complementary sometimes, right? And they also have their own biases, or maybe the antibody is not perfect in all regions and so on. And so if you have more data, we really show you how, if you have more data, how to put it together. But if you just have one of these assays, you can do all of these steps. And I encourage you to also try that. I mean, that's not a problem. Well, Dynamite will tell you which TFs are likely to explain the differences in gene expression that you put into the system, so to speak. Yes.
And what if it compares with the expression data we have? So the expression data we have says that this gene is up, but TF1, and the relation of Dynamite says that TF1, for example, activates this gene. By looking at expression data, we found that this gene is going down expression. So there is a conflict. How would you act?
I didn't really get the question, but the accuracy is not 1, so there will be cases where the classifier says it's up, but it's actually down. Is that what you mean? Well, that's life. But it's not perfect. I mean, it's obviously a simplification, right? There are many things that regulate gene expression. If that's not the only reason, but obviously that's one of the reasons. The other thing is that these data sets are not perfect. There's many problems, right? Often you don't sequence enough. If you would sequence much more, you would be better at calling background and signal, really, as Matthias said. And so, I mean, getting one is unrealistic, right? I think it's way too complex.
So when you feed the ratios of your cell type 1 and cell type 2, do you take account of replicate data sets by having an error on the ratio or something like that?
We don't do this yet because people rarely have replicates, but I think it's something we should really investigate much more to incorporate this. It's the same thing as before.
If I have replicate data, what do I do?
Yeah, then currently in this, we're not taking care of this.
So I guess I could run replicate, run Dynamite on all pair combinations and see how the results vary?
Yeah, that's one thing you could do.
So I have a second question as well, which is what happens when you have how does your tool cope with the cases where you have a transcription factor that upregulates some genes and downregulates other genes?
Yeah, that's a problem, right? So I've not really seen this, to be honest, because you're focusing on a set of genes here that are differential, Yeah, because it's not really all the genes. But honestly, I cannot rule that out. And what's going to happen now is basically the model is going to decide for the one where it makes most sense, right? So basically, if it's up, let's say it's activating more genes than it's repressing, then we'll probably decide for the activating aspect. But actually, what's interesting now, this is what this plot is look for, because that's really showing each dot is a gene. I don't know if I said this clearly. So actually, you see examples where this doesn't look as nice as this, where you have, instead of, let's say, everything is red on one side of the line, you also have a lot of blue ones. Yeah? And then you can see that this is, also in these cases, often accuracy will not be as good. And then you already see that it's not really a black and white picture. Yeah? But then you need to do much more complicated modeling to understand what's really going on. I think there was a question over here. Yes?
Yeah, so in the beginning you said that the benefit of using the segmentation methods is that there can be several transcription factors within one peak.
Mm-hmm.
Can you track that back from, like, if the whole prediction to the whole model, whether the model really relies on like the combination of two transcription factors instead of just the top ranking?
So it's definitely possible to track that back, although the model will not explicitly tell you, right? But the model considers all TFs together. Yeah, so it does this, and if it happens, we're actually doing this. But this is a good point. We could also, in a way, try to provide a visualization for this if there's actually they actually occurring. Yeah. I think we should stop the question now. I think we should really go to the practical, that you have some time to execute this, and we'll just be going around and helping you.